member of our ISCB Board of Directors, so welcome to the meeting. Our very first speaker, with no further ado, is Felipe, I'll butcher your name, I'm sure, Llinares-Lopez, who's going to speak to us about genome-wide detection of intervals of genetic heterogeneity associated with complex traits. Felipe.
So good afternoon, everyone. I would like to present our recent work on genome-wide detection of intervals of genetic heterogeneity. This is a joint work with my colleagues from ETH. So here, by genetic heterogeneity, we imply the phenomenon that several distinct sequence variants may all give rise to the same phenotype. It's known that complex phenotypes are often affected simultaneously by multiple loci. Therefore, we believe that properly understanding genetic heterogeneity is of utmost importance in order to understand the genetic basis of these complex phenotypes. Despite this importance, state-of-the-art methods often suffer from important limitations. For instance, we could think of using single-locus GWAS methods. However, these approaches do not consider the combined effect of several SNPs, and as a consequence, they tend to lack statistical power whenever sample sizes are small or the number of SNPs which jointly affect the phenotype is large. An alternative could be to use gene-based tests instead. These approaches essentially check whether the number of mutations within a gene is statistically enriched in cases versus controls. While they have the advantage of taking into account the joint effect of several SNPs, they will miss any variants which are not located within a gene or nearby a gene. We could also think of extending these approaches and test all genomic intervals without limiting ourselves to only genes. However, this would result in too many candidate intervals to test, and we would have effectively to choose between either having too many false positives or lacking statistical power. Therefore, we believe that there is a strong need to develop new methods which can detect genetic heterogeneity. In our work, we suppose we are given binary sequences of SNPs, ordered according to their position in the genome for different individuals. These binary SNPs could either be homozygous SNPs, or they could also be the result of a dominant or recessive encoding. Here, 0 stands for the major allele, and 1 stands for the minor allele. We also suppose that for every individual, we have a binary case control phenotype. As you can see now, in the interval highlighted in blue, there are 4 markers which exhibit genetic heterogeneity, since all of them have essentially the same effect on the phenotype. In order to leverage this genetic heterogeneity, we propose the following approach. For every genomic interval, we will build a new feature, which we call a meta-SNP. This meta-SNP is defined as follows. If all genomic markers within the interval are major alleles, then the meta-SNP will have value 0. And as soon as any of the markers is a minor allele, then the meta-SNP will have value 1. Therefore, you can think of this meta-SNP as a sort of indicator of the presence of minor alleles within a genomic interval. As you can see, in this example, this leads to a perfect correlation with the phenotype. In more realistic scenarios, what we believe will happen is that if a genomic interval truly exhibits genetic heterogeneity and has some association with the phenotype, then by building the meta-SNP this way, we are somehow aggregating individual effects into a stronger, easier to detect signal. With this in mind, the goal in this paper is to find all genomic intervals for which the presence of minor alleles inside the interval happens to be statistically associated with the phenotype. Of course, after a rigorous correction for multiple hypothesis testing. As you might guess, the challenge here stems from the fact that the number of association tests we need to perform is enormous. In fact, the number of tests scales quadratically with the number of SNPs. For instance, if we had 1 million SNPs, we would need to test more than 500 billion intervals. This implies, first of all, a huge computational challenge, but it also poses a great statistical difficulty, because after correcting for multiple hypothesis testing, the resulting statistical power would be too low. In our paper, our main contribution is a novel computational method, which we call the Fast Automatic Interval Search, or FAISS algorithm, which is able to achieve the goal we just presented while solving both the computational and statistical limitations. In our work, we test the association of every genomic interval and the phenotype by building a 2x2 contingency table using the meta-SNP corresponding to the genomic interval and the phenotype. Based on this table, we will compute a p-value using statistical association tests such as Fisher's exact test or Pearson chi-square test. Of course, we need to perform one such association test for every genomic interval. In order to correct for multiple hypothesis testing, we could think of using a Bonferroni-like correction and set the significance threshold to be the target family-wise error rate, alpha, divided by the total number of intervals. However, as I mentioned, if we do this, since the number of intervals is so large, we will have a very low statistical power. To solve this problem, in this work, we used a relatively unknown approach, first proposed by Robert Taron in 1990, which exploits the discrete nature that 2x2 contingency tables have. The main idea is as follows. Since contingency tables are discrete objects, under the null hypothesis, the p-values are actually not distributed like a uniform between 0 and 1. Rather, they come from some discrete distribution. This implies that for every table, there exists a minimum attainable p-value, which is strictly greater than 0. In turn, it could very well happen that for some intervals, this minimum attainable p-value is in fact larger than the significance threshold. Now, if this occurs, by definition, then those intervals can never be significant. And this also implies that they can never cause a false positive. We say that intervals like these are untestable. Now, with this in mind, Taron's method is actually very intuitive. All it says is, rather than using a Bonferroni correction factor equal to the total number of intervals, we only need to correct for those intervals which are testable, because the ones which are untestable can never cause a false positive. In practice, the number of untestable intervals greatly outweighs the number of testable intervals, and therefore, Taron's method leads to a great alleviation of the lack of statistical power. However, the computational challenge still remains entirely, because in order to find out which intervals are testable and which ones are untestable, we would need to build all the 2x2 contingency tables. To avoid this problem, what we need is an efficient way to prune the search space. In our paper, the criterion we use is as follows. If we have an interval which is untestable, and the corresponding meta-SNP for the interval has most of its entries with value 1, then we can prove that all other intervals which contain it will also be untestable. Now, this is very vague. Of course, it's more rigorously stated in the paper. But for now, I just want to give some intuition of why this makes sense. First, suppose we have a given genomic interval. And now we expand it by adding new markers to it. Then what will happen is that the new meta-SNP can only have the same or more entries with value 1. And to see why, it's actually quite intuitive. Since the meta-SNP just indicates if there are minor alleles within the interval, by making the interval larger, the number of individuals for which there are mutations within the interval can only increase. Now, the second fact is that when the meta-SNP has many entries with value 1, the corresponding 2x2 contingency table is going to be very skewed. Now, Taron proved in his paper that having a contingency table which is very skewed tends to result in the table being untestable. If we take an untestable table and now we make the meta-SNP have even more entries with value 1, the new table is going to be even more skewed. And this will cause the new table to also remain untestable. And then we arrive to the pruning criterion we stated. Now, next question is, what happens if we need even more power? Then we could think of using permutation testing approaches, which, as you probably know, can be considered the gold standard for family-wise error rate control. However, in this problem, clearly, naively applying permutation testing is absolutely unfeasible computationally. Because of this, in our paper, we propose a second algorithm, which we call Feiss-Westphal-Young. This method shares the same ideas as Feiss, but with a different goal. Here, we want to speed up permutation testing in our problem. Compared to Feiss, Feiss-Westphal-Young enjoys increased power, but it's also slightly slower. So you can choose between the two algorithms depending on your needs. Now, for the rest of the talk, I would like to give an overview of the main results we obtained. First, we want to check the computational efficiency of our algorithms. For this, we simulated a dataset consisting of 100 samples, and a varying number of SNPs, shown in the x-axis of this figure. The runtime of different methods is shown in the y-axis in seconds and using a logarithmic scale. The first curve, in orange, corresponds to the baseline of applying Fisher's exact test to every meta-SNP, but without any form of pruning. If we use now our algorithm, FICE, shown in green, first you can see that we can achieve a speed up of 2 to 3 orders of magnitude. But more importantly, you can also notice that the larger the number of SNPs, the bigger the runtime gap between the two methods. In fact, empirically, FICE seems to scale only linearly with the number of SNPs, whereas the baseline scales quadratically. Next, we want to see what happens if we apply permutation testing to this problem. First, we run a permutation testing algorithm using only 100 permutations for all meta-SNPs without pruning. And in blue, you can see that the runtimes we obtain are already quite prohibitive. In fact, even if we are only using 100 permutations, which is very, very low number, for 100,000 SNPs, the method already takes more than 1 day. However, we can get exactly the same results if we use FICE-Westphal-Young, but much faster, 3 to 4 orders of magnitude improvement in runtime. And compared to FICE, it's only about 1 order of magnitude slower. Next, we want to see how our methods do regarding statistical power. In this slide, we show statistical power in the y-axis versus signal strength in the x-axis. The first baseline, shown in blue, represents a very naive method which applies single SNP GWAS using Fisher's exact test. Next, we apply Fisher's exact test, but to every meta-SNP. For now, we are not using neither pruning nor Tarrant's method. And we see that just by taking into account meta-SNPs, we can increase power quite a lot for high signal strength. This somehow indicates that our way of building the meta-SNPs is indeed effective. However, the most dramatic increase in power occurs when we switch to FICE. By using Tarrant's method, we can increase power greatly, especially for moderate to low signal strength. And if, as I said, if we are willing to pay a higher cost in runtime, we can increase power even further by using FICE-Westphal-Young, now shown in purple. Now I will move on to results on real-world data. We used a collection of Arabidopsis thaliana GWAS datasets taken from Atwell et al. Out of the whole collection, we selected 21 datasets consisting of defense and development-related binary phenotypes. This results in sample sizes which range between 76 and 177 samples, depending on the dataset. And the number of SNPs was around 200,000. All SNPs were homozygous because the plants are all of them inbred. We ran FICE-Westphal-Young since it has the highest power and is sufficiently fast for all 21 datasets. And we compare it with 2 univariate baselines. First, Fisher's exact test for every single SNP. And secondly, a linear mixed model which can compensate for population structure. Now we want to answer mainly 2 questions. First, can our method truly discover novel intervals which would be missed by using univariate methods? And secondly, how affected are our results by population structure? To answer the first question, we look at the aggregate of all results in the 21 datasets. Out of 217 intervals that our method found, we can see that up to 70% of them are novel in the sense that no single marker contained within the intervals could be identified using the univariate methods. Therefore, a large proportion of all our findings are actually it's not possible to recover them using single SNP GWAS. To evaluate the impact of population structure, we took the following approach. First, we try to identify which SNPs are most confounded or inflated by population stratification. We do this by looking at the set of SNPs which are significant under Fisher's exact test, but which are not significant using a linear mixed model, which can compensate for population structure. Once these inflated SNPs have been identified, we check how many of them are present in the intervals our method reports. And it turns out that in these datasets, only 15 intervals lie in that category, which corresponds to just 6.9% of all our results. Therefore, we believe that population structure is not heavily biasing our results, at least in those datasets. We also had a collaborator who is a plant biology expert annotate some of the most significant intervals we found. And it seems that plausible explanations for different intervals can range between true genetic heterogeneity to linkage to some known causal SNPs or structural variation inside the region we found. Now, to wrap up, we have proposed a new method which can detect genomic intervals which are associated with the phenotype while correcting for multiple hypothesis testing. If you want to try out the method, we would be very happy, and our code is available in our website. And as for future work, I would like to extend this to work with heterozygous genotypes and or continuous phenotypes. I think building population and structure correction within the method would also be a great improvement. And finally, the same idea could also be applied, instead of to contiguous intervals of SNPs, to groups of SNPs in gene pathways. So thank you very much for your time, and I will be happy to take questions.
All right, thanks for a terrific talk. We have time for a few questions.
Yeah. Have you considered using linkage disequilibrium information to constrain? Okay. Have you considered using linkage disequilibrium information to constrain the number of analyses do you?
Okay, so we didn't try to do that. So we only took into account linkage disequilibrium to try to see whether our intervals were just a result of linkage disequilibrium, in which case it's not really useful if you are just finding the same SNP over and over again. And we found out, at least in these data sets, in Arabidopsis thaliana, it's not the case. But we didn't think about using that as a way to actually reduce the number of tests. So I think that could be an interesting way to extend the method as well.
So is it correctly understood that your method actually breaks down for anything above a 2x2 continuity table, so you cannot take into account any other variables when doing the test?
Okay, so for now in this work, we are only working, as you said, with 2x2 contingency tables. But now also related to the issue of extending the method to account for population of structure, we are working on an extension of this framework, which by using the Cochran-Mantel-Hansel test, can take into account any number of discrete covariates for now.
Yeah, coming back to the previous question about linkage disequilibrium, in your meta-SNP approach, you don't take into account LD actually. Like when you are building the meta-SNP, you are not already fusing SNP which are in LD.
Okay, so yeah, this is related to what I said before. So in principle, if there is a strong LD, it could happen, in fact, that by expanding the interval, you are just adding a SNP which is very correlated with the ones you have before. And what it can happen sometimes is that when you find an interval, you don't really find one interval. If you find that interval, plus many others which are slightly shifted, the same version. And we have a post-processing step, which I skipped due to time constraints, in which what we do, sorry for this, it's quite late. Okay, here. So what we do, if we find a cluster of overlapping intervals, we aggregate all of them, and we report both the boundaries of the cluster and the actual most significant interval within the cluster. And this somehow damps the effect of what you mentioned.
Great. All right, let's say thank you one more time.